This lecture video is going to go over the reproduction lab, which is titled, Is My Baby Healthy? In this lab, we're going to be doing 3 different activities to look at the overall health of your fictitious child. We'll be looking at whether or not the chromosomal numbers are correct as they should be or aligned up as they should be. We will be looking at genes, specifically going back to our original scenario with hemophilia. We'll be looking at the 2 genes involved in hemophilia and going through the steps of protein synthesis to make sure that the genes are correct and that they are not mutated. And then finally, you'll just be doing a generic pedigree just to kind of see how we trace different traits through a generation. As we get started, you're going to want to have this document handy with you so that you can look through it as we go through the different PowerPoint slides to help you through. What we're really trying to do is figure out what can go wrong during early embryogenesis. So what can happen at the very beginning at that cellular level that could lead to different kinds of problems, future problems for your unborn child. And so our objective here is that we want to be able to analyze a gene sequence. So going through the steps of protein synthesis, we want to analyze a karyotype to look at the chromosomes of your unborn child, and then look at family history and how things are passed on from one generation to a next. We'll start with an amniocentesis. So an amniocentesis is a test that is done when an unborn child, they go in and they take the fluid out of the amniotic sac. So they just inject a needle through that membrane, pull out some fluid, and then the membrane actually just closes itself right back up. The mother would be asked just to take it easy for about 24 hours before she could resume normal activities. But what we're looking for is as that embryo or as that fetus is floating around inside that amniotic sac, it's losing skin cells. Just like you lose skin cells every day you're walking around, babies also are losing those skin cells into that amniotic fluid. And so by pulling that amniotic fluid out, we can separate out the baby cells, the skin cells specifically, and we can kind of spin them around and we can add some dye to them and we can actually look at or image the chromosomes. And by cutting out all of the different chromosomes, which they just basically show them on a screen now, and then they kind of move them around, we create something that's called a karyotype. This is a karyotype. So here's the pictures of the chromosomes. Here are 2 different karyotypes. This one got cut off a little bit. So let's kind of look at this bottom one because it's complete. So remember that humans have 46 total chromosomes or 23 pairs. Okay, so there's 23 chromosome positions. There's chromosome 1, chromosome 2, chromosome 3, 4, 5, all the way to chromosome 23. And each one should have a homologous pair, right? Because we got 1 chromosome 1 from mom, we got 1 chromosome 1 from dad, 1 chromosome number 2 from mom, 1 chromosome number 2 from dad. So we have 23 pairs for a total of 46 chromosomes. Now, if you look at the difference between this one up here, so let's just focus on chromosome 1 here in this picture and this one, okay, there are still, go back to the chromosome counting activity that you did, there's still 2 chromosomes. There's chromosome 1, there's chromosome 1, okay? Chromosome 1, chromosome 1. The difference is that up here, these have not gone through the S phase. So when they took the cells, they hadn't gone through the S phase yet of the cell cycle to give them another chromatid. So this has 2 chromosomes. Each chromosome has 1 chromatid. Down here, it's 2 chromosomes. Remember, count the centromeres. But each chromosome has 2 chromatids because it had already gone through the S cycle by the time we got a picture of these cells, okay? So 23 chromosomes here, 23 pairs of chromosomes here for 46 total, 23 pairs of chromosome here for 46 total. Now, in a human, a healthy human is going to have 46 total chromosomes, 23 pairs. Each chromosome, we received one from mom and one from dad. So again, this is chromosome 1. There are specific genes that are found on chromosome 1. We got one from mom, we got one from dad. Here's chromosome 2. We have 2 of them, one from mom, one from dad. And again, the genes located on this chromosome number 2 are going to be different than the genes located on chromosome number 1. Okay, so here's chromosome number 3, the pair. So the first thing that we can do when we look at a karyotype is we can just look to see if we have the right number of chromosomes. A couple things I want to point out here at the bottom. So here's chromosome number 23. Chromosomes 1 through 22 are called autosomal chromosomes, and they code for all of our traits and all of our genes that don't have anything to do with sex. Okay? The 23rd chromosome is called the sex chromosomes because it is going to determine whether or not that baby is a male or female genetically. And there are certain genes that are going to be tied to these particular chromosomes. Now, in the case of a female, we would have an X chromosome and we'd have another X chromosome. We would have gotten an X from mom and we would have gotten an X from dad. In this scenario here, we have a male. We got an X from mom because a mom can only give an X to her unborn child. Think back to meiosis. And then we have a Y here from dad. Okay, so we have 2 different chromosomes, not like up here where we have homologous pairs. We have potentially 2 different chromosomes for chromosome number 23. So if we look at the difference between an X and a Y chromosome, just as some FYI, X chromosomes are much larger. You could have seen that in the picture. They're needed for survival. There are roughly 900 to 1200 genes that have nothing to do with actually female sort of sex characteristics on it, but that are needed to survive. So hemophilia, or the 2 clotting factors involved in hemophilia, they are found on the X chromosome. There's only one gene on this whole chromosome that really gives females their femaleness, for lack of a better word. Some of the other life necessary functions that the X chromosome controls are blood cell formation, kidney function, muscle nerve interactions, and then, like I mentioned before, blood clotting. So if you are a female, you are an XX. You have at least one X chromosome. If you are a male, you are a XY. You still have at least one X chromosome because it is needed for survival. The Y chromosomes are significantly smaller than the X chromosomes. They hold less genes. There's only about 70 to 300 genes on there, and almost all of them are genes that give males their more masculine characteristics. So there are no genes on the Y chromosome that are needed to survive, and that should make sense if you think about it, because a female is going to have an XX and they can survive just fine because they do, even though they don't have the Y chromosome. So there's no genes needed on the Y chromosome to actually help it survive. So when you look at your karyotype, you want to look down here and see, do you have 2 X's? That would indicate a female. Or do you have an X and a Y? That would indicate a male. So let's go back to your assignment for a second. So like I said, you need to take this sheet down here and you need to cut all of these chromosomes out. And then you're going to pair them up. Okay, so find the pair for this one, say right here. Once you get your pairs, you're going to put them in order from biggest to smallest. So the largest chromosome, let's just say it's this one, this would be chromosome number 1. And so you would have this chromosome in the number 1 spot plus its matching pair. Okay? And its matching pair is down here. So you have this one here and this one here, which are a match. I'm not entirely certain if if that one is the biggest one or one. It looks like it might be, so that would be chromosome number 1. Okay, so you would put those together. So once you have these all aligned, the first thing I want you to do is look to see, do you have an X and an X, or do you have an X and a Y? That's the first question that you need to answer up here. What is the sex of your unborn baby? Is it a male or is it a female? Then I want you to look at the karyotype, and I want you to count the chromosomes, okay? Do you have a situation where you have 47 chromosomes, or do you have a situation where you have 45 chromosomes? How many chromosomes do you have? Because by counting the chromosomes, we can figure out whether or not you have a genetic disorder. When you have a different number of chromosomes than 46, we call this an aneuploidy, and it refers to the idea that there is a wrong number of chromosomes. This happens during meiosis, and it's called non-disjunction, when the pairs don't separate out the way that they're supposed to. So instead of the one chromosome 1 going into one cell and one chromosome 2, or the other chromosome 1 going into another cell, they both go into one, and the other cell ends up not having any chromosomes, okay? So there are two types of aneuploidies. There's a monosomy, which means you're missing one of the homologous partners. So if you look at this karyotype down here, this is considered Turner syndrome. It's a monosomy because it's missing one of the 23rd chromosomes. It's missing either an X or a Y down here. Or you can have a trisomy. FYI, Turner syndrome is the only chromosomal monosomy that is survivable. If you look at this one, on this next one on this karyotype, you will notice that chromosome number 21, there's 3 of them. So this chromosome actually, this baby actually has 47 chromosomes versus 46. This, if you have a trisomy, depending on where it is, some of the more common ones are trisomy 18, trisomy 13, and trisomy 21. Trisomy 21 is Down syndrome. So Down syndrome is, if you've ever known anyone who's had Down syndrome, it's a disorder. But at this point in time, in our medical advances, a child with Down syndrome can live a very happy, fulfilling life. Okay, there are going to be some difficulties along the way, but it's definitely survivable, and survivable to a point where, you know, they can have a relatively normal, healthy life with experiences that, you know, a person with 46 chromosomes can have. So now if you have trisomy 18, that's a very different situation. You might not survive more than 2 days after birth. Okay, trisomy 13, that's going to be a different situation too. So depending on where the trisomy is, will determine the impact that it's going to have on that unborn child. So again, look at your chromosomes on your sheet and count the chromosomes. Do you have 46? And if you do not, if you have either one too many or one too few, explain what your situation is. And then you're going to have to do a little quick research on the internet. It doesn't have to take very long, like trisomy 21. What is that called? What are the symptoms? What's going to happen to your unborn child? Okay, so kind of pause this video at this point and then take care of doing that part of the assignment. All right, so let's move on to part 2. We discussed earlier when we started this unit about the idea that you're thinking about wanting to start to have a family, but one of your mothers has hemophilia, and the incidence of the disease can be traced back to different generations. So is it possible that your unborn child could have hemophilia? So what is hemophilia? Hemophilia is a change in either one of 2 genes or both of them. So there are 2 genes involved in hemophilia. The F8 gene, which is if you have a mutation in the F8 gene, that's called hemophilia A. A mutation in the F9 gene is called hemophilia B. So the F8 gene provides instructions for making a protein called coagulation factor 8. That's why it's called F8. It's a related protein called factor 9 is going to be produced from the F9 gene. And both of these proteins then are going to be involved in your body's ability to clot blood, or to basically form a plug in your blood vessels. So mutations in either one of these genes can lead to an abnormal version of either coagulation factor 8 or 9. Okay, so what you're going to be doing here is we're actually going to be thinking about what you did during week 1, during protein synthesis and DNA, is that you are going to be looking at, you are given, okay, this is the normal DNA sequence for F8. Okay, this is the DNA, TAC, CAG, GAA, okay? So what you're going to do is you're going to convert that then to a messenger RNA, right? That's the first step in making a protein synthesis. So you're going to change, go down the sequence. So T becomes A, A's become U, G's become, or C's become G. You're going to do that all the way down. So you're going to use this chart, right? We had AUG, remember? So let's just kind of write that here. So we would find A along the first, this is the first letter. Find the U is the second, so we know we're going to be in this box. Find the G over here, kind of go over, and it's called start, or methionine. So we would come over here, and we would put start. That's the amino acid, right? So then you do the next two. So CAG, right? So CAG would be G, U, C. And then come back down here. Look for the G is the first one. Okay, U is the second, so we're going to be down in here. And then C would be right here, so that would be valine. Right? We did this last week, or in week 1. Okay, so you're going to keep doing that for each group of 3 letters. Okay, each codon. So what you will end up with is the normal amino acid sequence. So a normal F8 amino acid sequence would be a start, a valine, something, 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 right? Now you're going to do the same thing, but this is your baby's DNA sequence, all right? So we were able to take a blood sample, again, through an amniocentesis, and we were able to get the sequence of the F8 gene of your child. So you're going to, again, make a messenger RNA, okay, use the chart then to put the amino acids in place. And you're going to look for errors, okay? Is this normal? First of all, you're going to want to, there are 3 possible errors here, right? You could have a substitution, and that's going to be where one of the nitrogen bases here on this DNA is going to be swapped out for another one. So you would compare this, the normal DNA sequence, to the baby's DNA. If one letter is swapped for another, that's called a substitution. You could have an insertion, or you could have a deletion. So an insertion is going to be when another letter is added in, and a deletion would be when a letter has been taken out. So once you figure out where your error is, then you're going to make your RNA again, just like you did up here, so opposite letters, and then use the chart to put in this amino acid sequence, and then down here you're going to put down what, if there's an issue, so it might be perfectly fine, or you'll put down, let's say it's a substitution error, and the result is instead of a valine, we have aspartic acid, okay? And thus the protein is faulty. And if the protein is faulty, if it doesn't match up here, this normal amino acid order, it's not going to work, and that baby would have hemophilia A. You're going to do the same thing down here for the F9 gene. Okay, so your child could have an F8 issue, they could have an F9 issue, or they could have both. So that's what you're going to try and figure out in this part here. Once you are finished with that, so again, you can pause your video and kind of finish that section. You're going to look at a pedigree. Okay, a pedigree is basically a family tree, or it's a tree to trace a gene as it is passed down from generation to generation. So when we look at a pedigree, what we look at here we look at some shapes. Okay, so typically males are going to be square. Okay, and females are going to be circles. If a person has a disorder, whatever disorder you're looking for, so let's just say we're looking at hemophilia here, right? Okay. The box or the circle will be completely shaded in. This person has hemophilia, this person has hemophilia, this person has hemophilia, this person has hemophilia, okay? If they do not have the disorder, then their square or their circle would not be shaded in. And on some pedigrees, it will show you a heterozygous situation, meaning that they're a carrier. So this person would be big H, little h, meaning that they don't have the disease, but they carry the allele for the disease. Now, on your pedigree that you're going to be working on, they will not be shaded halfway for you, so you're going to have to make some guesses. So we use pedigrees is to help determine whether or not a gene is dominant or recessive, whether or not it's sex-linked, and we can also use it to make predictions about genotypes. So if you go back to this assignment here, here's how this is going to work. I'll kind of get you started, okay? We know that hemophilia is a recessive sex-linked disorder, okay? Here is we can start with this male right here. They don't have hemophilia because they're not shaded in. So here's what its genotype would look like. Remember, it's an X-linked trait, so just like the Punnett squares we did in week 1, right? So, and we have a mother here who also does not have hemophilia. So we know that she's XX, and we know that she has at least one capital H, right? Because she does not have hemophilia. So, capital H is normal, and a little h would be hemophilia. Although remember that it had to be little h, if it was a female, okay? Now, the Y doesn't have any H's on it because the hemophilia gene is only found on the X chromosome. So how do we know whether or not this one is big H or little h? Well, we have to kind of move forward a second, okay? Let's look at these two parents' children. They have one daughter who does not have hemophilia. They have one son who does have hemophilia. So we know that that son is an X little h, Y. And then they have one son here that's normal. So we know that that son has to be X big H, Y. Okay? So here's the deal. Dad can only give this son his Y. That's the Y can only come from the dad. Right? So this Y ended up here. Which means that they had to get the little h from the mom. So this has to be little h because she passed this one on to her son, giving him hemophilia. So again, now go down here. We know that this is a male with hemophilia, right? So this would be X, Y, little h, right? So if you have a male with hemophilia, you would have X, little h, Y, okay? A normal male would be X, big H, Y, okay? And then females, if you put females over here, a normal female could be XH, big H, or they could be big H, little h, but this would be a carrier because they still carry that gene. So even though this mom is normal right here, she can pass this recessive trait on, and it's going to affect her sons in particularly because there's nothing to overcome that. And a female with hemophilia would be X little h, right? That would be with hemophilia. So now using this information, you're going to go in and you're going to fill in these genotypes, okay? And sometimes the easiest thing to do is for every circle, go put the 2 X's in right away, right? You know that there's going to be 2 X's here, and you know for every male there's going to be X and a Y. And then you can go in and fill the H's in as needed. And again, you're just going to kind of look to see how traits can travel through different generations. This question is individual 2. It's asking for the genotype, and then it's asking for whether or not they're hemophiliac, a carrier, or a normal. When you are finished with this activity, you can submit it. And then when you submit it, you can just like one sentence about the overall health situation of your unborn child. So for instance, my unborn child is a female with a normal karyotype, but a substitution mutation in her F9 gene, leading to hemophilia B. Okay? Hopefully this was helpful for you. If you have questions, please do not hesitate to ask.